Okay, so welcome back, my friends. Today we're going to talk about the importance of the niche for stem cells. So in the last lecture we talked about the research of Till and McCulloch and how they were able to isolate stem cells in the spleen of a mouse and able to tell that those stem cells were indeed responsible for the formation of colonies. Today, we're going to talk about the niche for stem cells and why that's important and how you form it, and just go through one example of hematopoietic stem cells and their niche, as well as an artificial niche that we kind of create within the kidneys of a mouse, and the interface that allows the formation of both bone and marrow cells. So let's start by getting a definition of hematopoietic stem cells a little bit, and then we'll go into the two different niches. So, a HSC. For those who don't remember, hematopoietic stem cell, and that is the primary stem cell for the blood. Right? Blood stem cell. Right? Now, here are the characteristics of the hematopoietic stem cell. First one is it's an adult stem cell. Right? So it exists in adult beings. It is not an embryonic stem cell. Quite straightforward. It is not pluripotent. Not pluripotent. Not pluripotent. And of course that means that it can't give rise to all three germ layers and all kinds of tissue in the body. It can only give rise to blood kind of tissues, blood lineage tissues. They're rare, okay? So only, there's not a whole lot, and in terms of refining them, it's very difficult to purify and culture these cells. The difficulty of culturing is another one of these points, but rare, right? There's not a heap of them. It's hard to get a hold of a good number of them. It's hard to proliferate them in an in vivo situation. And it's hard to get a lot of them for therapeutic purposes. Right? There's not a lot of them. And then there's hard to culture. Right? Why are they hard to culture? Well, they're non-adherent. That's an N. Non-adherent, right? They're non-adherent. They have a very specific niche. Which we're going to come to next. They are self-renewing. Which should be pretty obvious because it's a stem cell, right? And perhaps most challenging and interestingly, and as I've alluded to before, they're regulated by, and they regulate, the stromal tissue in the niche. Right? Right? So that means, and it contributes to this hard to culture point, right? They have a very specific niche, right? So they are regulated by stromal tissue in the niche. They're also, well, the stromal tissue, which is just connective tissue, which includes the osteoblasts and the other stromal cells in the bone marrow niche, makes it very difficult to culture these cells in vivo. Right? So now let's have a look at what that niche looks like very quickly. Need my rubber, need my razor. So what does that mean? Well, let's take an example, right? So there have been experiments done when we've taken not mesenchymal, but myeloid cells, myeloid progenitor cells, right? And then also within the bone marrow, there are fibroblast-like cells, right? Right, fibroblast-like cells. Okay? Now, if you culture those two separately, you get not much. But if you put them under the kidney sac, kidney space, kidney niche, kidney, just say in the kidney, in the area immediately around the kidney, you put a little nick in the kidney, and it's all kind of furry under there. And then you put these cells in, and it's just a nice way of localizing them. Also, it vascularizes the tissue quite nicely. You put these two in there, and you get bone and marrow tissue when they're together, and they're in a nice kind of environment, right? If you put them separately, you get nothing. This was a kind of high-level experiment, and it reflects the real-life niche of these cells. So let's have a look at the real-life niche. So these are osteoblasts, right? I've just taken a bone, right? And this is our, well, let's say it's actually like this. This is our cut here, right? So we're taking a little edge of the bone, and indeed we're looking inside the bone marrow, right? So these are osteoblasts. Right, each of those osteoblasts doing its thing. In addition, we have quiescent HSCs. Quiescent HSC, right? So these are quiescent hematopoietic stem cells. And we also have active HSCs, which are a little bit further out. They're the same size. Active HSC. Now the point we're trying to get at here is that this little niche, and we also have, I'm running out of colours. We also have stromal cells, which look, they're a bit elongated. Stromal cells. And I'm going to throw this marker in the bin. So we have our stromal cells. This is the niche within the bone marrow. Now the important aspect of this is that both these osteoblasts, and the HSCs, they both produce N-cadherin, which you can think of as like a pair of little gluey hands. So the molecule can be thought of as interlocking like this kind of deal, right? And so when both the hematopoietic stem cells, right, and the osteoblasts emit this N-cadherin, they adhere together, right? And that is one of the primary aspects of the niche, because they are able to adhere to each other, stick together, and form a nice little environment for the HSCs. In addition, the stromal cells emit cytokines, which we'll go through in another lecture. They emit cytokines and signaling proteins and all kinds of stuff. So these things together, and the osteoblasts also do their own emitting, and there's the blood cell, and there's a whole bunch of stuff going on, and they emit signals to each other. And this is the niche for the HSCs, and that is why it's difficult to culture HSCs, because they're not really adherent cells, they're kind of semi-adherent cells, which adhere only to the N-cadherin of the osteoblasts. And in addition, they need signaling from the stromal cells in order to behave properly. And that's why it's hard to culture these in vivo, and that is the environment and the niche for HSCs. Thank you.